Good afternoon and welcome to Biology Weekly. On today's episode, our hosts, Shanman, are going to be giving you guys a better understanding of what proteins are and why they matter. Proteins are one of four of life's basic building blocks, making them extremely important molecules to all forms of life. Today, we're going to be breaking up this video into 4 parts so you can have a better understanding of proteins. First, what proteins are made of. Second, how proteins link together. Third, the levels and bonds responsible for protein structure. And last, the functions proteins serve in our bodies. So proteins are molecules called polymers. Polymers consist of repeating units of monomers. In the case of proteins, these monomers are amino acids. So amino acids, the monomers, are the building blocks of polymers which make up proteins. These amino acids have a very specific structure and look something like this. They are made up of an amino group, a carboxyl group, an alpha carbon, and a variable R group. The amino group, circled in black, is composed of 2 hydrogens bonded to a nitrogen. Next is the carboxyl group, containing a carbon double bonded to an oxygen and an OH. In the middle, in red, is the alpha carbon bonded to a hydrogen. And lastly, we have the variable group R, which varies between amino acids and gives them their function. There are 20 different types of amino acids, highlighted here, required for human life to exist. And these different amino acids can create hundreds of thousands of possible combinations. The amino group, the carboxyl group, and the alpha carbon are the same for all amino acids. So the R group really is responsible for the variety among proteins. The first row of amino acids, highlighted, are nonpolar and therefore hydrophobic, meaning they do not interact with water. The second row of amino acids, highlighted, are polar and charged, meaning they are hydrophilic and love water due to the presence of oxygen and the CH bonds. Each of the 20 amino acids has a different variable R group, as I stated before, which affects how the protein folds. And changing just one amino acid can disrupt the shape and therefore disrupts the function of the protein.
Before we talk about how proteins link together, we are going to differentiate proteins from polypeptides. So first, a polypeptide is a chain of 3 or more amino acids that are not yet folded. Whereas a protein is when the polypeptide has taken on its full 3D shape. And so, in order for amino acids to link together to create proteins, a condensation reaction takes place. This reaction occurs when the OH from the carboxyl group of one amino acid bonds with the H of the amino group of another amino acid to form an H2O molecule, which is then eventually removed. Again, this process is also known as the condensation reaction. And so, once the H2O is removed, there is a covalent bond between the carbon and the nitrogen, which is also referred as the peptide bond, as circled in this picture. So, when 2 or more amino acids link together, we call this a dipeptide bond. And finally, when 3 or more amino acids link together, this is called a polypeptide bond.
Next, we're going to talk about the levels of protein structure and the bonds that hold them together. There are 4 levels of protein structure. First, we have the primary structure, which is the order or sequence in which the amino acids are arranged. These amino acids bond through peptide bonds. Next, we have the secondary structure, which is the shape of a segment of the polypeptide. There are two types. First, the alpha helix, which is observed on the right. Next is the beta-pleated sheets, which fold on itself on the left. These bond through hydrogen bonds. Next, we have the tertiary structure, which is the 3D shape of the entire polypeptide. You can note here that it is a combination of alpha helixes as well as beta-pleated sheets. The tertiary structure takes these alpha helixes and beta-pleated sheets and allows them to fold into a three-dimensional structure. Most proteins take on a globular structure once folded. These bond through hydrogen bonding, ionic bonds, van der Waals interactions, and disulfide bridges. Lastly, the quaternary structure, which is when more than one polypeptide chain interacts to make protein. They have the same bonds as tertiary structure. All proteins have primary to tertiary structures, but only some of them have quaternary structures.
Chaperones are proteins that assist in protein folding. When a polypeptide is being made, it goes inside chaperones where the amino acids can form proper interactions. Denaturation happens when proteins unfolds and loses its functions. This happens due to very high temperatures where the hydrogen bonds can break, and it can also happen in very acidic or basic environments. This is very dangerous because the shape of the protein is critical to its function. Functions proteins serve in our bodies. So the shape of the protein is crucial because it's directly related to its functions. One of the functions protein has is structural support. Keratin protein is an example of this, and this is found mostly in our hair and in our nails. Next, we have transport molecules. So protein is a major element in transportation of certain molecules. Hemoglobin is an example of a transport molecule because it transports oxygen throughout our body. Next, we have defense against disease. So proteins form antibodies that help prevent infection, illness, and disease. Following, we have the catalyst of biochemical reactions. Enzymes are proteins that increase the rate of chemical reactions in the body. Also, we have proteins that send and receive signals. So receptors respond to chemical stimuli outside the cell.
So that was this week's episode of Biology Weekly on understanding proteins with your hosts, Shannon. We hope after this video you guys have a better understanding of proteins, their structures, the bonds that hold them together, and the functions that they serve in our human bodies. Thank you very much for listening.